So, welcome everybody to today's research seminar. Of course, it's the first of our Genomics England research seminars after the summer break, so welcome back. My name is John Griffin. I am a scientific manager at the research management team in GEL, and I'm really honored to host you all today and to host the speakers for this seminar. Before we get started, there are a couple of kind of housekeeping things that I should mention. The first is that the seminar is being recorded. We always record the seminars and then we post them on the Genomics England YouTube channel. So just so you're all aware of that, and hopefully you're all okay with that. And by the way, all the seminars are on the YouTube channel, and there's a lot of really, really interesting talks there. So if you haven't had an opportunity to go and look at these talks yet, I would really recommend that you go and check them out. The other thing that I should mention is that it's wonderful if you want to chat and post questions during the talk. There is a chat box at the bottom for kind of general comment amongst the audience and just chat as we go through. And then please keep your questions specifically for the speakers to the Q&A box that you'll see slightly to the right end of your screen. And we'll try and get to those questions at the end, and we'll answer as many of them as we can. We have two really exciting sounding talks today. The first is going to be by Dr. Kristina Ibanez-Garikano, who's going to talk about the genetic prevalence of repeat expansion disorders in the 100,000 Genomes and TOPMed projects. Then after that, we are going to have Professor Holm Uhlig, who is going to talk about variants in inflammatory bowel disease. So I'm really excited that we're going to hear from these talks today. Before we do start, though, there's just one or two other things I want to kind of quickly draw your attention to, in particular some upcoming events that might be of interest to you. The first is our next research seminar. Of course, we have these every month, so the next one is going to be the last Tuesday of October, the 25th of October. And we have a really, really interesting lineup there. So Dr. Hao Monk is going to talk about the penetrance of PRNP genes. And Rachel Lennon and David Sherwood are going to talk more about their discovery pipeline for investigating the biology of basement membranes in disease. So I think that's going to be a really, really interesting seminar, and hopefully you can come back for that one in a month's time. The other thing that might be of interest to you is the Research in Genomics Equity Conference, which now has a new date. For those of you who might not know yet, it's going to be on the 20th of October in London. And this conference really aims to kind of discuss and explore all of the kind of recent advances and cutting edge developments in the area of data diversity. And so it really has a really interesting program, a lot of different talks and a lot of different discussions around that topic, including an opening keynote by Alice Popejoy. So if this is a field and a topic that you're interested in, then you might want to register for this. It's going to be a really, really great conference, I think. I think there's still spaces left, and you can register through the Genomics England website. I think Sam is going to put the link in the chat right now. So that's really, really exciting. The final thing then that I should mention is that we have a favor to ask of people who are RE users who use the research environment. We're currently running a survey where we're trying to get feedback on the RE from users. And so we would really appreciate it if you could take just a few minutes to answer that survey. And it really is an opportunity for you to help us shape the research environment and improve the research environment going forward so that it's better for everybody and better for research. So if you can take just a couple of minutes to do that, that would be really, really great. So I think Sam is also going to drop that link into the chat now for those of you who are interested. Okay, I think that's enough for me. I'm going to just then introduce our first speaker today, who is, of course, Kristina Ibanez. She is I guess, computational or computer scientist. Her background, her PhD is using bioinformatics and machine learning tools and developing machine learning tools to study, in particular, she developed machine learning tools to study the kind of comorbidities that exist between certain types of cancers and certain CNS disorders, and came up with some really, really interesting findings there. After her PhD, she went on to do a postdoc where she developed more tools again for analyzing gene panels and whole genome sequencing data before she actually joined Genomics England as a rare disease analyst, a senior rare disease analyst, and was instrumental in developing our pipeline for analyzing short tandem repeats. And actually that was later implemented across the GMS. So it was a pretty big contribution. She is currently head of computational genomics at Nostos Genomics, where she again is working to develop a lot of different tools for interpreting variants and ultimately improving interpretation and disease diagnosis. So we're really, really excited to have you here today, Kristina. And with that, I'm going to hand over so you can tell us more about this genetic prevalence of repeat expansion disorders in the 100,000 Genomes and TOPMed projects. Take it away.
Thank you very much, John, for introducing me. Can you see my screen?
Yeah, I can see it.
Okay, thank you very much for introducing me, and thank you also for everyone that is attending the GEL seminar. I'm going to walk you through the work that we are wanted to publish in, which is the analysis of the genetic prevalence of repeat expansion disorders within the 100,000 Genomes project and TOPMed. So the outline and summary of the talk is going to be, first I'm going to introduce what repeat expansion disease are, then the principal objectives that of this work, presenting you the cohorts we have been using, 100K and TOPMed. I will present you the methodology flowchart that we have followed up, and following the results and conclusions and some take-home messages. So first of all, what repeat expansions diseases are. These diseases affect usually the central nervous system and are caused by short and repeat expansions, or STRs, and as I'm going to tell in the call. STRs are units between 2 and 13 nucleotides that are repeated. So in this case, for example, we have the CAG block repeat, and it is repeated 7 times. But sometimes these CAG repeats become larger and larger and might cause repeat expansion disorders. There are over 40 repeat expansion disorders, and they are affecting different genes in different locations, exonic or intronic, and they have different repeat motif and also repeat size, so 3 nucleotides or even 6. And the symptomatic repeat, so the repeat size from which the repeat becomes pathogenic, varies across different diseases and genes. For example, Huntington disease, beyond 36, it becomes premutated, and from 40, we can already say it is pathogenic. So one of the main features of STRs is that they are highly mutable, and STR gradually becomes unstable and expands across generations, from normal to intermediate or premutated until full mutation or or pathogenic. So the next become more molecularly unstable, and they further expand in successive generations. It's been demonstrated, for example, for Huntington disease, that new expanded alleles arise from normal alleles with CAG repeat number that is in the high range for normal individuals, that in the high range for individuals that have been affected. Also, the allele frequency and the repeat size is inversely correlated or related. So larger repeat sizes are less frequent, and shorter repeat sizes are more frequent. As an example, here it is frequency distribution for Huntington, HTT. In the y-axis, we have the frequency, and in the x-axis, we have the CAG, so the motif repeats. So, and in green, yellow, and red, that you cannot see here, it's normal, intermediate or permutation, and full pathogenic cutoffs. So as I was saying before, larger repeats are less frequent. We have less frequency in population, and shorter repeats are more frequent. The epidemiology of these repeat expansion diseases varies. Usually, the majority of them, they are rare. So they are one per 100,000 genomes or individuals, sorry. And some of them are very frequent. For example, Thenajor, the frontotemporal dementia, Huntington, HTT, or myotonic dystrophy, which is the MPK. They are the most common ones that have been published or clinically observed. And also, there are some that are very rare, ATN1, DRPLA, or JPH3, but they are very frequent in some specific populations. For example, JPH3, the Huntington disease like 2, it's more frequent in Africans, or ATN1 has been seen mostly in Japan, in East Asia. The epidemiological studies are limited by clinical observation and genetic testing. Clinical observation based on the people that undergo genetic testing, or the presence of a particular neurological symptoms, or that there is someone in the family having a repeat expansion disorders. And it is also limited to specific populations. So an analysis that has been done in the northwest of Spain or another part in Japan. And these usually underestimate the true prevalence of repeat expansion diseases because of a misdiagnosed patient. Also, genetic when talking about genetic testing, usually capillary and gel electrophoresis or southern blots for larger repeats are being used as gold standard. And so far, and usually the large scale genetic screening is very expensive. And so far, the largest study to date, it's done by Gardiner in 2019, and it comprises over 14,000 of individuals, that mostly all of them are European descent, so white people. So large scale epidemiological studies are lacking for these repeat expansion diseases. As I was mentioning about techniques, the gold standard are gel electrophoresis, fragment analysis, and southern blots. But from 2016, more or less, thanks to high throughput sequencing, there are many bioinformatics software and tools. And at the end of 2016, we did a benchmarking in Genomics England, and we chose Expansion Hunter to detect short and repeat expansion from whole genome sequencing. And this year, in March 2022, we finally published a paper presenting the pipeline for detecting short and repeat expansions from whole genome sequencing, with a very large clinical validation that is now being used in the NHS. And this is the motivation from which we started working on this project. So I wanted to show you first this swimlane plot from the Lancet, which in the y-axis we have the genes that we included to detect, and in the x-axis we have the repeat size of these repeat expansion diseases in base pairs, in nucleotides. In this vertical line that I'm going to mention now shortly, it's the read length, which is set up to 150 base pairs. So more or less, Expansion Hunter accurately estimates exactly the repeat sizes for the majority of genes here. But for FMO1, as we were mentioning in the Lancet paper, since the blue, so the blue, yellow, and red background colors stays for normal, pre-mutation, and full expansion of pathogenic threshold. So repeat sizes that are beyond the read length, we cannot distinguish whether they are pre-mutated or they are full pathogenic. So that's why in this study, we are excluding FMO1. But for the rest, we can use Expansion Hunter repeat sizes by visual inspection, as I'm going to show you now. So the main objectives of this project are three. One, analyze the overall carrier frequency across these 13 diseases. So here I excluded FMO1, but we are including the Huntington disease like 2, JPH3. Also analyze the relative carrier frequency in order to answer the question, are there some genes that are more frequent in a particular populations? And also analyze the repeat size distribution across these diseases to answer if there is a repeat size, if there are some genes or some genome populations, that are enriched or differently distributed in some genes. How are we going to do that? By using genomes from the 100K and TOPMed projects, and also including the 1000 genomes project for control analysis. So just by a context that you might know, the 100K we have thanks to the Genomics England in the UK, and the main objective was to incorporate genomic medicine into routine care in the NHS. And we have for that 100,000 genomes in rare diseases and cancer. TOPMed stands for Transomics for Precision Medicine, is an initiative for the National Institute of Health in the United States, and the objective was to elucidate risk factors for heart, lung, blood, and sleep disorders, and for which we have over 53,000 genomes. And the 1000 Genomes project, the phase 3, that's why I put P3, it's another project from the United States, and it's a catalog of common human genetic variation. They are all controls. And we have 2,500 genomes. And the interesting thing here for the 1000 Genomes project is that they are homogeneously distributed across ancestries. We have 250 genomes per ancestry, per Europe, America, Africa, and so on. And most interestingly, both programs, 100K and TOPMed, contain individuals from diverse background with deep phenotyping data, and also, technically speaking, they have been sequenced in the similar way, 100 base pair reads, sequenced at Illumina high-seq, and with a depth coverage of 35x approx. So this is the methodology flowchart that we have follow up. First, selecting the genomes here, then estimating the ancestry of each genome. Then we genotype the disease loci per genome. And then we have done two things: analyze carrier frequency and analyze the repeat size distribution. So first of all, selecting the genomes, we have selected unrelated genomes. Neurological are excluded, we are not including them, and they have been sequenced in 100 base pairs in Bill38. The total number of genomes, over 82,000. We have inferred ancestry in the same way as in the Genomics in the 100K. They follow the same strategy in TOPMed, and these are the numbers. As expected, both cohorts and the total cohort is EU-centric. White people are enriched, it's decompensated, followed by Africans, Americans, South Asia, and East Asia is the with less people. So after that, we run Expansion Hunter version 3 across all genomes and across all 82,000 genomes. And we visually inspected all the repeats that were beyond the pre-mutation threshold for each gene. So I'm going to show you now the results of the carrier frequency analysis. So this is kind of forest plot. In the y-axis, we have the overall carrier frequency per 100,000 individuals per AR per each gene, and they are split by TOPMed in gray and 100K in black. The x-axis is the overall frequencies per 100,000. And we can see that the most frequent diseases that have been observed clinically, TNIORF, DMPK, and HTT, they are still very frequent in both TOPMed and 100K. On the other way around, the genes or diseases that are very rare, ATN1 or ataxia 7, JPH3 and TVP, they are also very rare, just on the left-hand side. But interestingly here, what it is very sexy from this data is that we have for AR and SCAS, so ataxia 1, ataxia 3, and CACNA 1A, which is SCAS 6, we are having more frequent carrier frequency than has been observed so far. So if I am putting here, enriching this in red, the reported carrier frequency per 100K, we can see that except ATN1 and ataxia 7, which are very rare, the vast majority of genes that we are analyzing in TOPMed and 100K are being more frequent than observed before in any clinical paper. This is the same thing, but quantitative with quantitative values. So the most frequent ones, TNIORF, Huntington, and DMPK, we can see that observed the known disease prevalence was high, but we still have high or frequent, sorry, but more frequent than expected. And then the rare ones are still rare. And the interesting things are the SCAS plus androgen receptor, which is Kennedy disease, which is basically male disease. And they are more frequent than expected. This is overall. What about relative prevalence? Is there any population that is enriched in a gene that it's more frequent in that population? So as we were observing, as it has been observed before, in Africa, JPH3 is prevalent, even though it's not, very rare in the general population, in the same way ATN1 in Japanese population in East Asia. But we can also see CACNA1A, SCA6 as frequent in East Asia, and DMPK in America and Europe. The relative prevalence, but just the other way around. So genes that are frequent in a specific population, ATN1, as we were saying, it's America, it's East Asia, sorry, JPH3, it's Africa. And the interesting thing here is that the SCA2, which we are seeing that it's more frequent than it has been reported so far, it's evenly distributed across all populations. So partial results of this first analysis is that we have seen more frequent prevalence to what it has been reported and published so far, particularly in SCAS, ataxia-1, 2, and androgen receptor. And we also have seen a slightly similar overall carrier frequency in both 100K and TOPMed genome projects using the 95% of confidence intervals, and also population-specific genes that are also similar in both datasets, JPH3 for Africans and ATN1 for East Asian or Japanese. So the second part of the work that I wanted to show you is the analysis of the repeat size distribution across populations. And coming back to the plots that I showed you in the introduction, it's been shown that repeat expansion mutation arise from a relatively large number of repeats in the normal range. And what we want to investigate now is whether there is differences in the repeat size distribution across populations. So this is results for 100K, the androgen receptor gene, AR. On the left-hand side, it's the distribution frequency. On the y-axis, we have the allele frequency, and on the x-axis, the repeat size. And on the right-hand side, it's a violin plot. And we have here the populations, European, East Asian, South Asian, America, and Africa. And on the x-axis, the same thing, repeat sizes. They show the same, but in a different way. So what I wanted to show you here is that here in looking at the violin plots on the right-hand side, we can see two peaks here in the European. And we can see that the first peak, it's the similar in American, East Asia, South Asia, and European. And the second peak is in red arrow. What about in Africa? In Africa, this peak is shift to the left, plus with two repeats shorter, so towards shorter repeat expansions. We can see here the same. Maybe some people prefer this, some people prefer that. So here we have the second peak of European, so this one, and we can see that Africa is here and European and the others are here. I am also here including the Wilcoxon p-value adjusted by Bonferroni, where African is significantly shorter compared to the rest of the Americans, of the population, sorry. What about TOPMed? Are we seeing the same pattern? So we reproduce the results here. This is the second peak. This is the first peak. This is the peak in European, American, East Asia, and South Asia, and this is Africa. And here the box plot, you can see that it shifts to the left with -2. What about 1,000? I like, as I told you, 1,000 genomes project, phase 3. It's interesting because all European, American, East Asian, South Asian, and European are homogeneously. We have 250 genomes per population. So when plotting, it's super interesting and super clear to see that all the population, except the Africans, stay here, while the Africans are shifted to have shorter repeat sizes. And here, for example, in the right-hand side, in the violin plots, we also have subpopulation information, so Yoruba, Sierra Leone, and we can see that all of them, even though breaking down by subpopulation, we can see that pattern, which make us see that they have repeat sizes that are shorter than comparing to the rest of the populations. Another gene that is affected is ATN1. So here is the peak of everything that is not Africa, and here is Africans. Here we can see better. I really like these violin plots because we have different shapes, let's say, but there is a main peak in all populations, European, East Asia, South Asia, America, and here is the peak of Africa. For quantitative values, I'm putting here the p-value corresponding to Africa, which is significantly smaller by 2 repeat sizes compared to the rest. TOPMed, again, we can see that shapes are different, but the first peak, the largest peak corresponding to each population, Africa is shorter. And sorry, and what about in, sorry, here. Well, sorry, so this should be AR, but I don't know why it's 81. So what about Huntington? So Huntington, we can see the same peak corresponding to all population except African. African are shift. Here we can see shapes are not the same, but the peak in Africa is here, while the peak on the rest are +2 repeat sizes. And again, here the p-adjusted value. In TOPMed, we reproduce the results of African being smaller, and on the 1000 Genomes project, we can see the same, even clearer, as I told you that because it's more homogeneously distributed. So partial conclusions of this second part of the work is that we see slightly similar repeat size distributions across all populations for 9 out of 13 that I haven't shown you, but it is the same. I just wanted to concentrate and focus on the African repeat expansions that are shift, plus with -2 or 3 repeat sizes to the left, which means having shorter expansions compared to the rest of the continental groups for AR, androgen receptor, ATM1, Huntington, HTT, and TBP. And these results are reproduced in the 1000 Genomes project phase 2. Take-home messages that I would like you to take. We've seen that overall carrier frequencies are similar in both large genome projects, 100K and TOPMed. And we have seen that they are more frequent than reported and published so far, particularly SCAS, ataxia 1, ataxia 2, ataxia 6, which is CAGNA1A, sorry, SCAS 6, CAGNA1A, and androgen receptor. And the true disease prevalence reported so far might be underestimated, maybe due to underdiagnosis, or maybe because there is not this kind of initiative of large projects. Possibly this is the largest cohort analyzing repeat expansion diseases, genetic prevalence, and looking at different populations. And last, we also seen that this pattern of having shorter repeat sizes in African genomes compared to the rest of the ancestries. I haven't done this by alone. I've been working together with Ariana Tucci in Genomics England and with some colleagues at the Bioinformatics Research in Genomics England. With TOPMed, I've been working with Bharati, Alejandro, or Alex, and Andrew from the Icahn School of Medicine at Mount Sinai in the United States. And at UCL, I've been working with Matteo Azzanovello and Valentina Galassi. And also I would like to thank Genomics England Research Consortium and all participants of the 100,000 Genomes project, and all of you that you have joined this Genomics England seminar. I'm keen to have any questions that you might have. Thank you very much.
Thank you, Kristina. It was a really clear and really fascinating talk.
Thank you.
Let's take a look. Maybe we have time for just a very quick couple of questions from the audience. So let's see, Deb, does penetrance kick in for African ancestry at the same point as other ancestries, or lower?
At the same point then. Penetrance. Well, I don't get this question. Penetrance in the sense that full mutation. So what we are seeing here is that it seems that African genomes, they tend to have less Huntington disease, or these 4 diseases that we have seen, that they are shorter. So doesn't mean that the repeat size that from which we say that they are pathogenic, it's shorter. We are saying that they tend to have shorter repeat sizes, so they tend to have less, they are less affected by this Huntington disease. But the cutoff, or the permutation cutoffs or full mutation cutoffs are not changed. It's the same. I don't know if this answers the question.
That's great. Okay, thank you. So let's see. Seema asks, is reverse phenotyping indicated to determine if some of the carriers are actually affected individuals?
Where are you reading this? Is reverse phenotyping indicated? Okay, to determine if some of the carriers are actually yeah, this is a good question. So so the carriers, yeah, so the thing is that we are taking carriers as not affected ones. So people that carrier and they might inherit their carrier if they have children or whatever. But we are not doing reverse phenotyping for this case. I will say that.
Okay. And then let's see, we'll take one more quick one. Let's see, maybe Edward Blair. Is the higher than reported carrier frequency telling us something interesting about our assumptions about penetrance?
Yeah, maybe. I mean, it opens a Pandora box. This is the interesting thing here. So far, all the published and clinical observed things are, I don't know, a Japanese family. They are like small. They haven't used whole genome sequencing, and I think that this analysis is the first of the many that will come. No? And so yeah, it opens a Pandora box, and it will change a lot the penetrance and the values that we are playing now.
Okay. So, yeah. Well, thank you, Kristina. We're going to move on and to the next talk. These questions will stay here, though. So if you have time, you could maybe just type some answers to some of them. We'll leave them here. And I apologize to people who we didn't get a chance to get your questions right now. So we'll move on then to our second speaker, Holm Uhlig. He is a professor of pediatric gastroenterology in the translational gastroenterology unit at the University of Oxford. And his research really looks at the genetic and immunological basis underlying inflammatory bowel disease. And really, I mean, if you want to get a sense of just the kind of impact that his group has had in this area, you can just, even if you take a quick glance at the publications from his group and the journals that they publish in, you'll get a good sense just of how influential their work is. And I guess an overarching aim for their work would probably be to kind of introduce clinical genomics into kind of standard treatment for people with inflammatory bowel disease, and also then to take and to translate what we learn about disease mechanisms from these studies and translate that into actual useful treatments. So I won't say much more about that, but I will hand over to you, Holm, and thank you very much for coming and talking to us today.
Thank you so much for the invitation to speak at the seminar, and it's a great pleasure because it's this research environment and the ecosystem that we use, and where so many collaborators of us collaborate jointly. I like to discuss inflammatory bowel disease and give one exemplar for a single gene variant and one copy number variation where this research environment has contributed. So inflammatory bowel diseases are a group of heterogeneous disorders. Most prevalent are Crohn's disease and ulcerative colitis, with a prevalence of about nearly sort of 0.5% in the Western country populations, and it's a disease of young adults typically. But in addition, there is recognition that there are unusual patients with extreme phenotypes, extreme early onset of disease, or concomitant primary immunodeficiencies, and they have an increased frequency of monogenic defects, orphan disorders that can disguise as inflammatory bowel disease, but actually sort of are a different genetic makeup. And prototype of such a disease group are the IL-10 signaling defects. They typically present with a few months of age, and this is a group of disorders where either macrophages and monocytes cannot respond to IL-10 signaling, or macrophages or regulatory T cells do not produce IL-10. And this is a group of disorders that are fatal if not treated, and hematopoietic stem cell transplantation is now a curative approach in these disorders. However, this is not the only group. There is now an emerging spectrum of diseases, and there are other groups that act in the phagocytic department. Then there are immune dysregulation disorders that affect regulatory T cells, or epithelial defects. And this overall shows that there is a complex mix of now more than 100 genetic disorders that can present as intestinal inflammation, and that complement our knowledge on these complex polygenic diseases, Crohn's disease or ulcerative colitis. It typically starts with one patient, and the example I want to give is a patient that was initially started by Ying Huang from Shanghai and my colleague Alexio Muis from Toronto. It was an infant that started with severe inflammatory bowel disease, with skin lesions and with signs of arthritis, didn't grow very well, and was having massively raised inflammatory parameters for over 2 years, and then sadly died under the age of 3 from this severe disorder. Immune workup in this patient did show that there was an exceptional increase of a Th17, Th1 response, and our lab is studying many of those rare patients, and this is sort of an exceptional presentation with extreme high numbers of ROR gamma T positive IL-17 or IL-22 positive cells in the peripheral blood. Alexio Muis then identified by exome sequencing a monoallelic variant in the spleen tyrosine kinase in this patient, and shortly after, by coincidence, found another family in Toronto with a variant at the same position, even if it's a slightly different amino acid exchange. So it's a serine 550 tyrosine or phenylalanine change. And this is interesting because this Syk kinase is downstream of a number of immune relevant receptors, for instance TCR, Fc gamma receptors, or dectin or CLEC receptors that are all important for immune responses and pathogen responses. Now, functional analysis of these variants did show that this is a gain of function variant, and there is an increased phosphorylation of this Syk, and the functional experiment suggested that this is an autophosphorylation event that is increased, and several assays by Western blot or by FACS could show that there is a dramatic constitutive activation of this Syk despite the absence of ligands. So this would fit with an autosomal dominant and single monoallelic disease. Now, if you have one patient or two, then there is always a question, is this sort of the right variant? And so we were very fortunate to collaborate with Dali Li in Shanghai, who made a mouse knock-in variant that replicated the human amino acid exchange, and these mice developed very, very severe spontaneous arthritis, and Dalili could show that this is an exceptional auto-inflammatory process with very similar findings that Dominic Aschenbrenner had shown sort of in this patient from Shanghai. And so there was now sort of not only sort of a replication in 2 families, but in a mouse model as well. At this stage, we reached out to many colleagues throughout the world, including the Genomic England consortium, and we collaborated with Sophie Hamilton and Helen Griffiths and Karin Engelhardt in Newcastle, and we identified several additional patients, including 1 patient from identified in jail. The overarching phenotypic finding was that all of these patients had intestinal inflammation. A large group of patients had skin inflammation, joint inflammation, and lung inflammation. 2 patients had central nervous system inflammation, and 1 had liver inflammation. All had recurrent infections, all had hypogammaglobulinemia, and 2 patients developed lymphoma as well. So overall, there were now 6 patients with variable age of diagnosis. This age of diagnosis correlated with the autophosphorylation defect in vitro, so that did make sense, but there was a clear similarity in the presentation, and the mouse model replicated this. So overall, this group of sick gain of function variants explained the disease by an hyperactive autophosphorylation event. The mouse model suggests that hematopoietic stem cell transplantation is an curative treatment for these patients, and this is currently followed up by several groups. Similarly, we could show in vitro that a Syk inhibitor, fosfarmatinib, blocked the autophosphorylation event, and similarly sort of this raised a question that is biologically very, very relevant. What are the drivers of this disease? So when we did an analysis of the intestinal microbiome, we could show that these patients who sadly died had high numbers of Candida tropicalis in the stool, and although we cannot be sure at the moment whether this was the driving factor, shortly this would have been a modifier of the disease, because this is upstream of C-type lectin receptors and would further aggravate this disorder. These studies are only possible by collaboration, and I already mentioned Sophie and Helen, Karin and Suzanne, who contributed to the Genomic England analysis, Alexia Muis in Toronto, and Ying Huang, who looked after the initial patient. Our lab here in Oxford collaborated with Khan, Bostock, and many other collaborators, and the study has been published last year. Now, we are not only interested in the monogenic diseases, but the question was, are there any other drivers for unusual features? And so we performed a follow-up study looking for chromosomal numeric aggregations and copy number variation, and again with the Genomic England Research Consortium contribution. This study did confirm by a literature review that in Turner syndrome, several markers are very clearly hinting to an increased prevalence of inflammatory bowel disease in the region of about 2%. That is statistically significant, but relatively low prevalence. All other disorders that we know that are associated with different chromosomal numeric aggregations do not seem to be associated with intestinal inflammation. We then looked for copy number variation and compared the published literature with copy number variation that is available in databases, for instance in Decipher. And this is very, very difficult because there is no pattern emerging. So these seem to be individual patients. The reports that have been published in the literature are difficult to reproduce. There are some monogenic disorders where deletions do play a role, causing haploinsufficiency, for instance deletion of CTLA4. Homozygous genes can be identified by genomic analysis, for instance, loss of a copy in XIAP in males, or compound variants that cause recessive disorders. One of the copy number variants caught our attention because it's a potentially testable hypothesis, and this was a patient that was most recently identified with an increased copy of the IL-2 receptor, and this has been published just last year, and that is an interesting locus in the IL-2 and IL-15 receptor, and the authors around Nadine Serf-Bensusan could show that there is an functional effect, and they speculate that an increased stimulation by IL-2 by 3 copies in the IL-2 RA locus would drive a pathogenic response. So we looked in multiple data sets, including the Genomic England dataset for patients with IL-2 and IL-15 duplications, and indeed we could identify a patient that was almost sort of identical in the size that was duplicated, as well as in the age of onset, extreme early with pancolitis, no immunodeficiency, but difficult to treat. And we could show functionally that this variant did show an increased surface expression of the IL-2 receptor, and that is almost a replication of a very rare phenotype. However, the question is how to deal with conflicting evidence. Because when we looked at the 100,000 Genome Project and Decipher and ClinVar and a number of other databases, then copy number duplications in this locus are described, and we have no evidence that they all lead to intestinal inflammation. So we rather see this variant still as a variant of unknown significance. It's highly interesting. There could be sort of additional contributors to the disease. We have some functional evidence that this variant does have an impact on the immune system, but that have 2 patients with an extreme unusual presentation, but there is conflicting evidence in the databases, and so this is an exemplar where we have still to do further research to see whether this is linked to a pathogenic response. If it is, then there must be additional factors that contribute to the disease. Now, to come up with an integrated picture of genetics in inflammatory bowel disease, Chrissy Bolton and Sumit Pandey and Chris Milley from our lab and from Boston performed an an analysis to review all available evidence for 165 disorders where intestinal inflammation had been described. We reviewed the likely penetrance, the clinical phenotypes, the expression on the gene and protein level, including single-cell sequencing and biochemical pathways, to build a model of this group of Mendelian diseases. This shows the penetrance in this group and the age of onset of the higher penetrant disorders. And as you see, typically age of onset for IBD is between 20 and 40 years of age, and the majorities of this disorder, more than 2/3, present substantially earlier. The large group of these disorders are expressed in the immune system, in particular in lymphocytes and in macrophages. There is some expression of some genes across different compartments, and here is a single-cell transcriptomic analysis showing the epithelium, the mesenchyme, the endothelial cells, and immune cells, and then there is a group of disorders specific to the epithelium as well. And when we put all these data, including biochemical data, into a system, then there is a pattern emerging with a very large group of phagocyte activation problems, T-cell selection and immune regulation problems, epithelial defects, an emerging group of endothelial expressed dominant genes, but then there is sort of another 20 to 25% of genes where we actually do not know. And I have to say this already has some clinical relevance, because in at least 50% we have now reasonable evidence that suggests that hematopoietic stem cell transplantation can be or should be considered in groups of those patients, and that opens up the way to precision medicine in these highly complex disorders. To summarize, we have now sort of a better insight into cellular mechanisms. It's partially linked to specific groups of the barrier function or immune responses, but a number of genes are expressed across different compartments, and we still lack clinical evidence how to treat some of these patients. In about half of them, as I mentioned, stem cell transplantation can be an option. So we are now in this phase where we sort of come from patient-centric research towards exploratory genetics. There is now a guideline from the ESPGHAN, the European Society for Pediatric Gastroenterology. We have just finalized a guideline for the UK from the British Society of Gastroenterology and the Pediatric Gastroenterology Society. And the next step is how to translate these findings back towards patients. But the last 10 years have seen sort of a major change, and this is really sort of from research into clinical practice, and this has been driven by studies such as the Genomics England project. With these were the three studies that I've highlighted, and this has been done by multiple collaborators in the UK and internationally, and these are the funders. Thank you.
Okay, thank you very much. It was a beautiful talk. Let's take a look for the questions. Okay. Let's see, first one here at the top. Can epigenetic mechanisms affect the development and progression of IBD?
Yes. So So I have focused on genetics and relatively high penetrant diseases. But what we have seen in the last 40 years is an several magnitude increase in prevalence, initially in Western countries, but increasingly in Asia and in developing countries as well. And that must mean that there are clear environmental factors, and there are increasingly indications that both in immune cells and epithelial cells, epigenetic mechanisms drive either disease susceptibility loci. So there is this interaction, and Jack Sansaki has published, for instance, that certain genetic GWAS susceptibility loci are affected by methylation changes as well. We have shown that certain key mechanisms can be affected, for instance, by metabolic factors from bacteria. So, for instance, butyrate can affect HDAC. And HDAC obviously sort of has an impact on histone, and environmental factors via epigenetic factors can drive both protective as well as disease-causing mechanisms. Smoking, cigarette smoking, is a clear susceptibility for Crohn's disease, and obviously there are epigenetic factors. So yes, there are epigenetic factors, and the important thing is to see this increasingly sort of from a genetic, epigenetic, and functional side.
Okay, yeah. We have a whole series of questions here from Isatu, but I think some of those might just be comments. So maybe the last question, I'm just going to skip down here to Stig. IBD is a frequent side effect of immunotherapy of cancer. Do you know of any association between genetics and therapy-induced IBD?
Yes, it's a brilliant question. So we see in the clinic sort of a revolution in oncology. That means that several sort of diseases that were untreatable, like melanoma, some other cancers, are now treatable, or at least it can be delayed or cured even by checkpoint inhibitors. And for instance, anti-CTLA-4, PD-1, and other therapies had revolutionary impact. Interestingly, sort of all these have equivalence in the genetics. So there is CTLA-4 haploinsufficiency, and the phenotype is very, very similar. So there's thyroiditis, hepatitis, enteropathy, colitis, dermatitis, and we see this sort of in the genetic field as well. So it's a bit proof of concept that the targets are indeed relevant. Now, there is increased, because this is an experiment of nature. You sort of have a treatment and you develop intestinal inflammation. Mm-hmm. So these can now be used to look, for instance, for interaction with the microbiome, because you don't have to wait for 20 years. You have basically an outcome after a few months already, and we can see who is responding, who might be more prone to side effects, and which type of side effects are happening. And that could be sort of even relevant for the genetics field, where this might explain why in 20 or in 30% of some genetic disorders, intestinal inflammation is present. How we potentially even can prevent some phenotypes, either in patients or more relevant in these checkpoint inhibitor treatments.
Okay, thank you. I guess we're basically out of time, actually. Yeah, sorry, I'm hesitating because I was actually going to ask you a question, but no, we'll let it go. I won't run us over. But thank you very much to both of our speakers for your time today, for coming and telling us about your fascinating work. And thank you for all the audience members who are coming too. I'm going to call it a day there, I guess, but we will hopefully see you all at the next seminar, which again is at the end of October. So thanks very much, and bye.